Okay, so good afternoon, everyone. So today we will be start ruminant Trichostrongyles nematode parasites. So I will take you in the world of ruminant livestock and their parasites. So today and tomorrow will be covered some of the most important Abomasum parasite for clinical point of view. So first thing, you have to use your point solution, and the password is this session ID is PTHP505. It should be same in all my lectures. So just put that one in your point solution first. And if it is working, Okay, so if it is working, let's see if it is working or not. Identify the nematode in the following images. Okay, so B. So these left, these are the flukes, these are the tapeworms, and B, these are roundworms. So we will be covered all these nematode parasites in next lectures, this week, next week, and week after the next week. Okay, so the learning outcome for this Trichostrongyles nematode parasite of Abomasum. So you will get the basic understanding of the life cycle epidemiology or the transmission of these parasites, and you will demonstrate the parasite gastroenteritis. This is PGE. This is a complex of one or two parasites together. You will get the knowledge of the abomasal or intestinal Trichostrongyles nematodes of sheep and cattle. You will work on the biology, diagnosis, and the major issues regarding the treatment plans. You will further analyze the epidemiology and disease pathogenesis. For example, what is the difference between clinical type 1 or clinical type 2 disease in Osteotagia. So you don't need to remember this taxonomy. The only thing you have to remember is now you are dealing with Trichostrongyles nematodes. And again, in the next lectures, we will be covered the most important Trichostrongyles of ruminant, Osteotagia, Tildocegia, Homunculus, Trichostrongyles, Coperia, and Nematoderus. However, Dictocallis you already covered with the Metastrongyle lecture with Kamal. And the predilection site of these Trichostrongyles in the case of the ruminants, in the case of the sheep, in Abomasum, Homunculus and Tildocegia is normally reside there. In the intestine, Trichostrongyles species like Veterinaris or Clubliformis are there, Nematoderus are there. So we will be covered mostly those who are in blue shade here. These are the most important one we will be covered in the next few lectures. And the case of cattle, Osteotagia, very important, Coperia is important, and Dictocallis is important, and you already covered that one. Okay, so Trichostrongyles nematodes, most important cause of production losses and economic losses in livestock. How? Due to the treatment and management cost, due to the death of the calves, lamb, or kids, poor growth rate, and these are most important cause of the production losses in livestock. So what happened when the conversion of these Trichostrongyles nematode from egg to larvae, that is normally happen in the posture, always in the posture. No transmission of these Trichostrongyles nematodes or parasites is happen in the confinement situation, because the animal must be graze on the posture to become infected with these Trichostrongyles nematodes. The reason is the infective larvae are always in the grass, not in the confinement situation. These parasites are not the soil transmitted parasite. These are transmitted when the animal is grazing on the pasture. So when we will talk about the life cycle, you will be easily understand why we are saying that these animals should be on the pasture if they are infected with these parasites. So young animals, because they are nourishing stage, they do not ingest these pasture because they are not in the pasture in the grass. They are normally in the confinement situation, and they will not be infected very quickly. So young animals do not ingest the level of forage until they reach into the weaning stage and go outside and do the grazing. So that's why you will not see the Trichostrongyles disease in a very, very young animal. Normally, you will see this disease in the case of sheep and goat. They will be around like 2 to 3 months of age, and in the calves, they will be around 3 to 4 months of age. Okay, so these Trichostrongyles gastrointestinal nematodes are the important disease syndrome of ruminant, and they caused by the nematode parasite of Abomasum as well as intestine. So we covered these abomasal and intestinal parasite next few lectures, and normally you will call them a Trichostrongyle nematodes. They cause the infection as a single species. Some of the species they cause the infection as a one single species, like Haemoncosis. One single dominated species cause the infection. But some of these Trichostrongyles species, they make a PGE complex, parasitic gastrointestinal complex, and the multiple species can cause infection together. In the case of small ruminants or sheep and goat, Haemonchus, Tildosagia, and Trichostrongyles. These are the most important parasite who will be covered. In the case of cattle, Haemonchus, Ostrategia, and Coperia. These are the most important parasite will be covered in the next few lectures. Okay, so the most important point is, without knowing the life cycle of these Trichostrongyles nematodes, you will not be able to understand the pathogenesis of this disease or the epidemiology or the biology of this disease. What I said before, animals should be in the grass to become infected. So if you don't know about the life cycle, you will not understand that concept. So the life cycle of these Trichostrongyles are always a direct life cycle. So how this life cycle work? Adult, they lay egg. Egg is come out along with the feces in the environment, and this will further, eggs will convert it into L1 and L2 stage larvae. These 2 stage larvae are always in the feces and in the egg shell. After that, see this posture. These larvae need this type of posture to become infected the host. So this L2 further converted into L3 stage larvae. This is the infective stage larvae, and that is normally seen in the posture and is a high in number, and that is the infective stage cause infection in the host. And this L3 stage larvae has a very strong cuticle outside that can protect that larvae from the environment. Once this goat or the ruminant graze here, they will take this L3 stage larvae from the posture, and then the L3 larvae goes into the predilection site. What is predilection site? This is the site where this parasite reside. Whatever we're going to talk about, like now we're going to talk about the Abomasum parasite today's lecture. So it means that this L3 stage larvae go into the Abomasum. So this, once they reach there, this L3 is further converted into L4 stage larvae or L5 stage larvae. These are the juvenile stage, and they are always within the host. So L5 stage larvae finally converted into adult. Once it converted into adult, they are sexually differentiated between male and female. So this is how the life cycle of this group of parasite work. Okay, so I really want to know that you understand the life cycle of this parasite. So now I'm going to ask you one question. Which of the following larvae is the infective stage of Trichostrongyles nematode life cycle? They reside. Yeah.
In like healthy dogs that reside in the somatic tissues, just there, what would be their predilection site?
Yes, you are right. So because they have a migratory behavior, normally they reside in the intestine, but they because they have a migrated behavior to move around into the other organs. So once they go there, but they come back again into the intestine finally. Right? So that's why the final destination is intestine.
Okay.
The parasite is belong to intestine, but they have a migratory behavior. That's the difference. Okay, so great. L3 stage larvae. So which of the following stage of Trichostrongyles life cycle infect definitive host? Okay, L3 stage larvae. Okay, so the seasonal pattern of the gastrointestinal Trichostrongyles nematodes. So let's see if A is April and M is May. So this is all year around. So normally the nematodes, we'll talk about that one in the intestinal, once we talk about the intestinal parasites, Trichostrongyles parasite, we'll talk more detail about all of these species. But in general, they normally, their season is on from May to July. Tildosagia circumcenta, they are from July to November, and Haemonchus is on the mostly in the rainy season. From somewhere this September to November. Not the rainy season. The tropical rainy season is a little bit different, but I can tell you why this one is a little bit different here. Trichostrongyles is normally in the winter time. So this pattern is more close to the temperate region. But you have to be very careful that in the tropical region, the pattern is a little bit different. Like, for example, the some of the tropical region, if you talk about Haemonchus infection, that is mostly in June and July. Somewhere the rainy season, July is the most rainy season there. But this is more or less toward the tropical areas like Canada or somewhere Scotland. So that's the difference you have to make it once you think about the seasonal pattern of these Trichostrongyle nematode parasites. So the clinical features of Trichostrongyle nematodes, the anorexia is the main hallmark of this disease in ruminants. Diarrhea is very common, hypoproteinemia, anemia, and the well-being is decreased. So these the clinical features are the general features of the Trichostrongyle nematodes. And also immunosuppression, that is one of the biggest clinical features in these Trichostrongyle nematodes. Other features are also there, like submandibular edema or bottle jaw, dehydration, poor odor, rough body coat. So these features are the general features. Once we will talk about each individual species, will be talk about what particular specific feature of that disease are. So which particular disease cause submandibular edema. So we'll talk about that one in detail once we come to the specific diseases in the next part of the lecture. So the diagnosis generally based on clinical sign or history. Fecal egg count, you will be perform the fecal egg count in not this week, after the next week. So some clinical pathology, you will be look it into if animal is become anemic or hypoproteinemic. Necropsy is also one of the way to look of these worm. You will not able to see all of these worm because some of them are very tiny to see by eyes, but some you can see it very easily, and I can show you some of the example where you can see the worms. If you open the abomasum, you can easily track that these worms, because they're a little bit bigger in size as compared to those are in smaller size. It depends which particular parasite, Trichostrongyle parasite is there. So the method for measuring the fecal egg count. So McMaster, you will perform the McMaster a week after the next week in the lab. Easiest and quickest method, and that method is very useful for the is a choice for the sheep and goat. You can this method is used to be look it into the sheep and goat poo. The detection sensitivity is 25 to 50 egg per gram of feces, and normally you will be see high number of egg count in the case of sheep. That's why the sensitivity is good enough to look it into sheep and goat. But the sensitivity is not good in the case of cattle, because normally the fecal egg count are low in the case of cattle. So that's why mini float is the best method in the case of cattle. This is recommended for cattle. You can detect like 5 egg per gram of feces. That is sensitive. Modified Wisconsin method is also there, but that method is little bit time consuming, and more is the precision is not very much accurate. That's why is not very much recommended method for different labs, but some labs, diagnostic lab, use this method. The sensitivity is between 1 to 10 egg per gram of feces. So what we're going to see once you will be looking into the fecal egg count. So this type of in the microscope you will maybe see these type of eggs. You will count them, yes, but Trichostrongyles, you can't be able to differentiate Haemonchus from Tildocegia. So we always prefer you to be use the word you are dealing with Trichostrongyles egg. As long as you will know that in this region haemonchosis or haemoncus is very common. So yes, this potentially may be haemoncus, or you can do the PCR, right? Other than that, you can easily differentiate this nematodarus. This is one of the trichostrongyle. That is the only one you can identify that yes, this is nematodarus and this is trichostrongyle egg. Other than that, Monasia, this is tapeworm, coccidia, that is protozoans. Maybe you will see all of these other trichoris there. So as far as trichostrongylus, you will be definitely say these are the trichostrongyle eggs, except nematodarus. So anthelmintics are sold in the market is for sheep and goat. They are always a broad spectrum nematocides, and we discussed about these nematocides in the last lecture. And depend on the class of anthelmintic, different administration route has been used. For example, in sheep and goat, oral administration is the best route, where benzimidazole is the choice. White wormer always come on the oral administration. Levamisole, these are membrane depolarizer or nicotinic agonist, oral or injectable. Marantol is normally come with the feed premix orally. Microcyclolactone, these are the ivermectin type of drugs. They come in pour-on or injectable form in the case of cattle, and oral in the case of sheep and goat. So pour-on microcyclolactone is the most common method used in cattle. However, that is one of the worst method to be adapt in the treatment of worms in cattle. The reason is, once you use as a pour-on, you will be the efficacy of these spread of these the drug in the body is not very good. So you will maybe find the drug resistance issue. Oral microcyclolactone are the most effective choice, but there is no oral product sold in cattle in USA and Canada. On the other hand, injectable is the much better choice as compared to pour-on, and the resistance is one of the huge problem in the case of sheep and goat, and one of the emerging problem in cattle. So you have to keep in mind once you go into the treatment plan, and we will be talk about more in detail in our control of ruminant parasite lectures somewhere week after the next week, where we'll be talk about more in detail what you will gonna do as a control strategies, because resistance is one of the biggest issue in the livestock industry. So now we'll be covered these most important genera of Trichostrongyles, but today we'll be just focused on Haemoncus. Is a barbel pole worm, highly pathogenic, most important parasite of sheep and goat in USA, and one of the important parasite of cattle as well. So Haemoncus, three most important species. Haemoncus canthatus, they are highly pathogenic, followed by Haemoncus placei and similes. These are the blood-sucking parasite, and normally the parasite of small and large ruminants, and the abomasum is the predilection site. This parasite is always reside in the abomasum, and is relatively a big parasite, 2 to 3 centimeter long, and you can easily, once you open the abomasum, you can easily see these reddish color parasite because of the blood sucking parasite. You can easily identify these parasites from the abomasum. The eggs are brown in color, and they have a blastomere inside. So again, I will be more prefer to say these are the Trichostrongyle eggs, as long as I am very much clear based on some other diagnostics that these are Haemoncus egg. So Haemoncus canthatus is the most important parasite as I said, sheep and goat, normally in the tropical and subtropical region. And this parasite has a minor importance in cattle, but can also be infect the calf. And the beauty of this parasite is they can switch the host, mostly a pathogenic from sheep and goat, but also infect cattle. And also the beauty of this parasite is they can infect together in the same host, like Haemonchus contortus and Haemonchus placei. Once they infect together, they will be mate between the species and produce the hybrids. So co-infection and production of hybrids are very common in this parasite. So the question is why these hybrids are important. These hybrids, the female hybrids, they are viable. And once they are viable, Haemonchus contortus is the highly resistant parasite in the world. And this is one of the route of transmission. The resistance genes are transferred from Haemonchus contortus to Haemonchus placei via these hybrid worm. So that's why this parasite is very, very clever to do these type of things without brain. Haemonchus placei, on the other hand, important parasite of cattle, but it can also infect sheep, most important parasite of tropical and subtropical region. And in recent years, there is a big issue of this haemonchosis in southern part of United States. So you will be see these cases in the southern belt in a daily basis. So very, very common there. So 500 worms are enough to cause a clinical disease. Why? Anyone? They are blood sucking. So that's why 500 is more than enough to cause the clinical disease. Pre-patent period is 3 to 4 weeks. So because they are filled with the blood, that's why we called it barbel pole, parents of this parasite, as shown here. Okay, so let me ask you one question, if your point solution is ready. So what is the most important name of Haemonchus? Okay, barbel pole worm. Okay, so the life cycle of Haemonchus. So my question is, the life cycle of Haemonchus is different from the life cycle we talked about in the journal Trichostrongyles nematode? How many of says yes? Is similar or is different? Similar. So who gonna explain me the life cycle of Haemonchus based on this one? Any volunteer? We discussed that one just like 10 minutes ago.
So it leaves the species
So,
and then it develops from L1, L2, L3. And once it's in L3 and it's in the pasture, the goat or sheep will eat it and then it'll go through until it's the egg
and then converted into adults. So these are the male and female adults. Great. So this is what I'm expecting from you to be understand the life cycle. Once you know that, you can easily understand the concept of this Haemonchosis disease or any other Trichostrongyles nematode disease. Okay, great. So for the epidemiology or the seasonal pattern of this disease, naive sheep, they produce the eggs in the spring or early summer. So now you know the life cycle. So it means that eggs are in the feces in the spring and summer. So what's going to happen? Egg is converted into L3 stage larvae. Yes, because they are in the environment in spring and early summer. And that goes much faster once they come into the environment in this type of posture. They will be, once the temperature increase, they will be start converted into L3 stage larvae. First L1, L2, and then L3 stage larvae. So this L3 stage larvae is on the peak somewhere here and then start dipping down and stop in the autumn because the temperature change. This parasite is the tropical parasite. They need that summer rainy season to convert into L3 stage larvae. However, again, as I said, this parasite is very, very clever without brain. Now this parasite is normally seen in the southern part of United States all around the year, not in any particular season, not in like seasonal pattern in that particular month. So that's the problem. This parasite is now emerging as a whole season parasite in USA, southern part of USA, like Georgia or Florida, these states. You will see this parasite all year around, right? So, as I said, this is evolved in the tropical areas, flourishing in warm weather, and also now we have seen this clever parasite in the colder climate region, where like Canada or Scotland or Sweden. So that is a temperate region, but we have seen this parasite in the summertime in these regions. So this parasite changed their genetics and evolved into these new regions. And the transmission is peak in the warm and wet weather. This is what we already discussed. This parasite needs a rainy environment to become better temperature to evolve. However, in the first lecture, I give you the definition of hypobiotic state or arrest state. Anyone remember that? Yes? This is the state or the condition when parasite goes into inhibit in the abomasum or wherever the predilection site is. In this case, abomasum. And what happened? This parasite, which stage of the larvae is infective stage and taken by the host? L3. So L3 is converted into L4 in the abomasum and stop there, not further converted into L5 and adult stage. So that is the state where this parasite come into the hypobiotic situation. However, why this parasite do that? Because the temperature outside is not favorable for this parasite to be convert the stage, produce the eggs, and eggs come out in the feces in the environment because they will not survive. So think about how clever this parasite is to know that what is going on outside, but the parasite is in the abomasum, but he knows that what is going outside. So he has to be stopped their conversion stage to go into the hypobiosis, and then once the conditions are favorable, they come out and change their stage. So this is how the concept of the hypobiotic state is. And keep in mind, always L4 is the stage where this parasite go into the hypobiosis. So we discussed this is the blood-sucking parasite, and the main feature of the clinical feature is the blood loss, because sucking blood 500 is cause a very severe disease. In sheep and goat, disease is really, really severe, and death are very common in sheep. If 5,000 or more than 5,000 eggs are in the fecal egg count is there, so the animal is really, really anemic. However, in the cattle is not very much severe. Sometime because the cattle is a bigger animal, the loss of blood is not easy. and the worm burden is not very much high as compared to sheep and goat. And not mostly, but most of the time, we will see the PGE complex. I told you about what is PGE. Is a parasitic gastroenteritis complex. It means that more than one parasites can cause infection in a one single host. So if you do the fecal account, and you will be see in the microscope, maybe you see some other parasite along with Haemonchus. So this is a PGE complex. But sometime in the young calves, Haemonchosis itself causes a very severe disease. That is also, I have seen a lot of cases on that single species infection of Haemonchosis, especially in the young calves. So the most important point as far as the clinical feature of this disease is concerned is anemia. Why? Sorry? Blood sucking. Second most important point is submandibular edema. We called it bottle jaw. So these are the two most important clinical features in this disease. Other than that, you will see some animal become weak, rough coat, weight loss, some black tarry feces, but you will not see any diarrhea. So as I said initially, Trichostrongyles clinical features. I talked about general features. Now I'm talking about some specific features cause clinical disease in sheep and goat or cattle in Haemonchosis. So these 2 points are really, really important. Just remember these 2 points. Anemia is very common when you look it into this mucous membrane, eyelid mucous membrane, and you will also see this bottle jaw. This is submandibular edema. So these 2 are the most important sign of Haemonchosis, and you can remember these 2 signs. So in the severe cases, as I said, if the fecal count is more than 5,000 or higher, maybe the blood transfusion should be used. If you will not, then animal maybe die. So the diagnosis, clinical sign and history. So based on the clinical sign, which 2 most important clinical sign you will gonna look? Exactly. So these 2 important points you will look once you go into the clinical examination. However, keep in mind, if you say animal is anemic, but sometimes anemia can also caused by some protozoan parasite or fissulosis. In the region where you work, you may see these other diseases may cause anemia. Therefore, fecal account is important. You have to perform the fecal account along with the Pharmacia score. So this is the Pharmacia card. We'll talk about more. I'll show you that one in our practical. So what is going to happen in Pharmacia? So these are you will put this card in front of the eyelid, and you can look it into this particular color of the eyelid. If the eyelid color is something like that, so it means that this is the situation of the animal, the treatment is needed. So it's based on the eyelid color in this Pharmacia card. So, which of the following statement regarding Haemonchosis is true? Okay, hypobiosis play important role in the epidemiology of this parasite. Okay, so now what are the most important clinical sign of Haemonchosis? Very, very important. Oh, sorry. stop here. Anyway, so anemia and submandibular edema. Okay, so what are the most common tests to diagnose haemonchosis? Famishaka. Okay, great. So the treatment. So treatment is very important because, as I said, anthelmintic resistance is one of the biggest problem in the livestock industry. Benzimidazole, microcyclolactone, the resistance is very, very common. Levamisole, the resistance is least common in Haemonchus, and the resistance is very, very high in Haemonchus contortus as compared to Haemonchus placiae. So what gonna happen? So what you gonna do? I mean, for example, if you treat the animals like in companion animal, dogs come into the clinic, you just treat them, deworming, job is done. Job is not done here. In the livestock, you have to think about, once you treat the animal, you have to perform the fecal egg count reduction test. It means that perform the fecal egg count before the treatment, perform the fecal egg count 14 days after the treatment, that your drug is working or not. That is really, really important. In the case of this resistance issue, always go with the use of different drug combination. Never trust on one single drug, because you never know which form is become infected, become resistant to which drug. If you go into the combination, you will be get the better results. So we'll talk about these combinations in the control strategies lectures. Okay, so these are some of the data in USA, the resistance prevalence. For example, if you look it into benzimidazole, is almost like at the level of fixation, 100%, 97%. In the case of ivermectin and moxidectin, ivermectin again is very high, and moxidectin is pretty much better, and levamisole is also in a better shape. So this is one of the old data, but now the resistance is much increased even in moxidectin, but levamisole is still working in most of the forms. Okay, so finally, these are some of the work I have done in my career, because my all career belongs to Haemonchus. I did a lot of research on haemonchosis and anthelmintic resistance. So these are some of my work on this group of parasite. Finally, so what do we do if the anthelmintic is no longer work? And this is the scenario at the moment. So anthelmintic drugs are not working. So these are some of the steps you have to adapt, like sustainable integrated parasite control parasite management. So this is one of the approach you have to adapt, and we talk about most of these points in our control of Trichostrongyles lectures. For example, how to improve the posture. We'll talk about that one in detail. Refugia management. What is refugia management? What is the targeted selective treatment plans? And the use of smart drenching, like use of anthelmintic wisely. Don't use the anthelmintic in a massive way, because these are very important resources. Anthelmintic drugs will come into the market maybe in years, like maybe 10 years. So use these product wisely. Vaccine, There is a vaccine of haemoncosis in New Zealand and Australia, barbel pole vaccine that is available there but not in USA, and some non-chemical integrated approaches and biological control. We already discussed about some fungi used as a feed additives. So these points most points will be covered in the control of Trichostrongyle lectures in the next coming days. New drugs, at the moment there is no any drug I know is available in the market, are going to come into the market. So the drugs we already have, the major groups we already have, those are the only drugs we have available to be control these parasites. So use these drugs wisely. Okay, so anyone has any questions? For one thing, I would always prefer, if you have any questions, sometime what happens if you have a question during the lecture, you maybe forget at the end. So I would suggest, please raise your hand once you have a question, even right into the middle of lecture. We can discuss that question and then move on. So anyone has any question? Yes.
So in terms of goats, what would you recommend as like a deworming preventative, just like your annual when you're working with cows or goats and you deworm them? What would you use? What would you recommend?
So what particular anthelmintics you are saying to be used? So exactly the same what you're going to use in sheep, because most of the drugs, like the anthelmintic drugs, labels are written there, used for sheep use only, but we normally use exactly the same in the goat, right? Benzimidazole, yes, you can use in sheep and goat. Mm-hmm. Yes.
I was wondering if you could clarify that you had mentioned that Poisson had thorns in cattle, but then there's not very common disease associated with cattle. Like, how is that?
So Haemonchus placiae, yes, it causes disease in cattle, but is not very severe as compared to Haemonchus contatus in sheep and goat, right? And secondly, this in cattle, sometime you will see the PGE complex, like parasitic gastroenteritis complex. Some other parasites can be infect along with Haemonchus placiae. So that's why the severity will not be so high if a single species infection can be there. Anyone has any question in their mind? No. Okay, thank you.